Assalamu alaikum, welcome to another YouTube video. In this video we're going through a chapter red blood cells through Guyton and Hall. If you have a textbook, so don't forget to go through the chapter. So let's start. First of all, erythro means red or cytes mean to make. The process which produces red blood cells is known as erythrocyte. RBCs develop from erythropoietic stem cells. Function of RBCs: they transport hemoglobin, which in turn carry oxygen from the lungs to tissue. Another important function is carbonic anhydrase. Carbonic anhydrase is an enzyme which catalyzes reversible reaction between the carbon dioxide and water, which form carbonic acid. The main important function of RBCs is acid-base buffering. The structure of RBCs. RBCs are biconcave discs having mean diameter of about 7.8 micrometer and a thickness of 2.5 micrometer at the thickest point and 1 micrometer at its center. The average volume of RBCs is 90 to 95 cubic micrometer. 100 milliliter of blood contains 34 gram of hemoglobin, and the amount of hemoglobin in men is about 15 gram per 100 milliliter and in female 14 gram per 100 milliliter. The next is production of RBCs, erythrocytes. Size and stages in fetal life include mesoblast, hepatic and melanoid. Mesoblast, early week of embryonic life in which RBCs are produced in yolk cell. Hepatic, middle trimester of gestation in which liver and spleen produce RBCs. Next is melanoid, last month of gestation in which bone marrow produce RBCs in human body. In children, all bones long plus flat produce RBCs in bone marrow, while adult 18 to 20 years red bone marrow produce RBCs. Red bone marrow is also known as active bone marrow. Stages of RBCs. It includes stem cell. Primary cell capable of self-renew and differentiating into specialized cells is called stem cell. Pluripotent hematopoietic stem cells go through committed stem cell, then produce committed stem cell and clone-forming unit stem cell. Lymphoid stem cell, which is again divided into T lymphocyte and B lymphocyte. CFUS divide into CFUB, CFUGM, CFUM. CFUB again divided into CFUE, which produce erythrocyte. CFUGM again divided into granulocyte and monocyte. CFUM again divided into megakaryocytes and platelets. The process of erythropoiesis include: number 1 is cell size, which is decreased 25 to 7.2 micrometer. Disappear nuclei and nucleus. Hemoglobin formation. Change in properties of cytoplasm. Mitosis occur at normal stages. Pluripotent hematopoietic stem cells formed colony-forming unit erythroblast. Next is proerythroblast formed. The other formation of polychromatoblast erythrocyte. The next form orthochromatic erythroblast, then reticulocytes formed, and finally erythrocytes are formed. Proerythroblast is about a start of erythroblast. First of all, the pluripotent hematopoietic stem cell is about 23 diameter, 4 to 5 nuclei present in it and deep basophilic. The next is proerythroblast, which is about 2 to 3 nuclei, fine reticulum chromatin, 20 diameter and basophilic. Polychromatoblast, which is early normoblast, intermediate normoblast and late normoblast. Early normoblast have 15 diameter and no nuclei. Intermediate normoblast about 10 to 12 diameter and hemoglobin start appear. At the late normoblast, hemoglobin and the nucleus disappear while its diameter is about 8 to 10. Reticulocyte. Reticulocyte is about 5 to 7.5 diameter and reticulum is formed. Erythrocyte is about 7.2 diameter and reticulum disappears. Factor of erythropoiesis. Erythropoietin is a glycoprotein that regulates the process of erythropoiesis. Regulation of RBC production. The first one is tissue oxygenation, and in which anemia, high altitude and diseases take place. Anemia when the decrease of oxygen of the tissue take place or increase in the RBC's production. High altitude, the quantity of oxygen is less in air Due to this, RBCs greatly increase. Disease. Circulation decreases tissue blood flow, which leads to the failure of oxygen. Thus, the production of RBCs increase and it may lead to the cardiac failure. Erythropoietin stimulate the RBC production, which increase in response to hypoxia. Hypoxia or the number of RBCs. About 90% erythropoietin is produced in kidney, while 10% is produced in liver. The renal tissue hypoxia lead to increased tissue level of hypoxia inducing factor 1, which serve as transcription factor for a large number of hypoxia including genes. Including the erythropoietin genes, HIF1 bind to the hypoxia response element, and in return erythropoietin gene inducing transcription of messenger RNA, hence erythropoietin is formed. Erythropoietin performs some functions which include it stimulates stem cells, promote hemoglobin synthesis, and promote the stages of erythrocytes, and also promote the release of RBCs. Special maturation factor, vitamin B12 and folic acid for the synthesis of DNA, formation of thymidine triphosphate. Thymidine triphosphate production depend upon vitamin B12 and TTP in DNA RBCs which cause its maturation. Thus, vitamin B12 and folic acid promote maturation of nucleus and cells. Deficiency of vitamin B12 and folic acid lead to cell irregular, large and fragile. Fragile lead to short life, about half-life of RBC, and maturation failure. Factor for hemoglobin include iron, copper, cobalt and nickel, and vitamins. Iron for heme formation, copper for iron absorption, and cobalt and nickel for utilization of iron in hemoglobin. Vitamin B2, B3 and B6 and C include. Thanks for watching my YouTube video. If you like this video, don't forget to subscribe my YouTube channel. Thanks for watching video.